Hello everyone, my name is Darshini Satyamurthy from DBT3A. My metric number is 23 DBT 20F1025. Today I am going to do a lab simulation based on bacteria enumeration. Let's go to the experiment. Click on the blackboard to view a larger board for discussion. This is the meaning of enumeration of bacteria. As part of daily routine, the laboratory microbiologists often asked to determine the number of bacteria in a given sample as well as having to compare the amount of bacteria growth under various conditions. Enumeration of microorganisms is especially important in dairy microbiology, food microbiology, and water microbiology. Knowing the bacteria count in drinking water, fresh milk, buttermilk, yogurt can be useful in many aspects of industrial microbiology. Bacteria are so small and numerous counting them directly can be very difficult. Some of the methods used involve diluting the sample to be point at which the number of bacteria has been reduced to very small numbers. Observe the three links given below to bring you to the virtual lab that I wish to perform. If you have performed all of the exercise, you can click on the end lab. Now I am going to do viewable plane count. Viewable plate count is one of the most common methods for enumeration of bacteria. Serial dilution of bacteria are placed onto an agar plate. Dilution procedure influence overall counting process. The suspension is spread over the surface of growth medium. The plates are incubated so that colonies are formed. Multiplication of bacteria on solid medium result in the formation of a macroscopic colony visible to naked eyes. It is assumed that each colony arises from an individual viable cell. A major limitation in this method is selectivity. The nature of the growth medium and the incubation condition determine which bacteria can grow and thus be counted. Visible counting measures only those cells that are capable of growth on the given medium under the set of conditions used for incubation. Sometimes cells are viable but not be culturable. The number of bacteria in a given sample is usually too great to be counted directly. However, if the sample is direct serially dilution and then placed onto an agar surface in such a manner that single isolate bacteria from visible isolate colony, the number of colonies can be used as a measure of the number of viable living cells in the known dilution. Normally, the bacteria sample is dilution by factor of 10 and plated on agar. After incubation, the number of colonies onto dilution plate showing between 30 and 300 colonies is determined. A plate having 300 colonies is chosen because this range is considered satisfy signification. If they are less than 30 colonies on the plate, small error in dilution technique or the presence of a few contamination will have a drastic effect on the final count. At the end of the incubation period, select all of the agar plate containing between 30 and 300 calories. Plates with more than 300 calories cannot be counted and are designed too numerous to count TNTC. Plates with fewer than 30 calories are designed too few to count TFTC. We will be testing 4 samples of water for the variable count. The first one is water from a drinking fountain, boiled water from a drinking fountain, water from the local river, boiled water from the local river. You will need to data 1 to input your data and calculate the number of CFU per ml. Take 6 dilution tubes, each containing 9 ml of sterile saline. Dilute 1 ml of a sample by withdraw 1 ml of the sample and dispensing this 1 ml into the first dilution tube. Using the same procedure, withdraw 1 ml from the first dilution tube and dispense into the second dilution tube. Subsequently, withdraw 1 ml from the second dilution tube and dispense into the third dilution tube. Continue doing this from tube to tube until the dilution is complete. Transfer 1 ml from each of only the last 3 dilution tubes onto the surface of the corresponding agar plate. Incubate the agar plate at 37 Celsius for 48 hours. Choose a plate that appears to have between 30 and 300 calories. Count the exact number of calories onto the plate. Click on the dilution tube rack of test tube to bring them to the table. Each of the dilution tube contains 9 ml of sterile saline dilute solution. Next, click on the water sample to bring the sample to the table. Now click on the eyedropper to withdraw 1 ml of sample. Click again on the eyedropper to withdraw 1 ml from the first dilution tube and dispense into the second dilution tube and subsequently withdraw 1 ml from the second dilution tube and dispense into the third dilution tube. Continue doing this from tube to tube until the dilution is complete through the dilution tube 6. Click on next when the dilution are complete. The dilution for each of the 6 dilution tube can be summarized in the image below. Next, we will inoculating agar plate with the last three broth culture dilution. Click on the agar plate on the shelf to bring them to the table. Now click on the eyedropper to transfer 1 ml of dilution 4 to plate, 1 ml of dilution 5 to plate 2 and 1 ml of dilution 6 to plate and 3. Next click on the pencil to label the agar plate. Click on next when finished. Click on the agar plate to place them in the incubator at 37 Celsius for 48 hours. We will now need to perform this same dilution and inoculation step for each of the test sample. The process is the same for each sample. We will assume the process of dilution and inoculation has been completed for all of the water sample and the 48 hours of incubation time has now been completed. Click on the incubator to bring all of the inoculated agar plate onto the table. Each of the group of inoculated plate is labeled with the source of their respective sample. Observe the three links given below to bring you to the video lab that I wish to perform. If you perform all the exercise, you can click on End Lab. Now I'm going to the End Lab. That's all from me. Thank you.